Hello, everyone. My name is John Myers, and I'm here to talk to you today about the spatial reach of brainwave synchronization. I would like to thank LabRoots for having me today, and I would like to thank you all for your attention. The first thing we want to talk about is to understand the underlying nature of brainwaves. We've all heard of these things, these spectral changes that happen inside the brain, but what are they? So brainwaves are electromagnetic waves that represent the flow of electrical currents through the brain, right? And these currents are very complex, so they can send detailed information across neural tissue. But why are brainwaves so complex? Well, there are billions of neurons, and each one has multiple electrical fields at the sites of the synapses. And you probably already know that neurons are connected to each other via synapses. And at each one of these locations, an electrical field is generated. When you add billions of neurons together, they get mixed together, and the signal becomes very hard to interpret for us on the back end. To record these brainwaves, we use many different acquisition tools, some being non-invasive and some being invasive. The non-invasive measures include magneto and electroencephalography, which are very similar measurement techniques, the magnetic signal being read by magnetoencephalography or MEG, and the electrical signal is read by EEG. You can see there in the bottom that these signals are very similar, so you kind of have a great similarity between electrical and magnetic fields, right? They're just pretty much the same signal. And so on the right, you see the invasive measures, and we see here that there are brain rates there on the left and brain waves there on the right. When an action potential occurs, they contribute to the brain waves that we read with these electrodes here. And our non-invasive measures, our invasive measures include electrocorticography and microelectrode arrays. Microelectrode arrays read single unit activity at a high density. So they contain hundreds of tiny needles or electrodes that read neurons, single cells. And these electrocorticography grids, the ECOG, those read grid-like brainwave activity from the surface of the brain itself. Now, these measures, these invasive measures obviously require neurosurgery, right? And so how do we interpret brain waves? Well, if we take an example here and we inserted an electrode into the monkey's auditory cortex, we can use analysis tools like this to understand the waves themselves. So if you zoom in on that little patch of cortex, you can see that each little dot is a recording contact. And if you play a sound, the next panel over in A there shows the actual brain waves itself. If you play a sound at this time 0, you can see the brainwave response to the sound in auditory cortex. Panel B shows us if we just make all the negative voltages one color and the positive voltages another color, we can see that there is a spatial distribution of these waveforms across the cerebral space. The final measure here is CSD, or current source density. This tells us how local the activity is. This gives us an indicator as to the current sources and sinks, or input and output of electrical activity at this particular location. And you can see here that across the layers of the cortex, that's 1, 2, 3, 4, 5, and 6, you can see that there are inputs and outputs, the sources and sinks, the positive and negatives, the blues and the reds. You can see different distributions of patterns there for those. So this helps us interpret a particular, these patterns of communication between brain regions. So these brain regions are synchronized, right? These brain waves are synchronized within this region. And that gives us an indicator as to the spatial distribution of that synchronization, like where the input is, where the output is, etc. Right? And so brainwave synchronization is also called coherence. The reason why we call it coherence is because the theory is that when a given population of neurons synchronize with another given patch of neurons or group, let's say cell group A and cell group E or B there, whenever they synchronize, they share information. So their membrane potentials can oscillate in a kind of song or a symphony. And when they're synchronized at the same frequency, it is theorized that they can convey information more easily across one another. B and C, for example, have a bad phase relationship where the peak of one wave corresponds to another wave. And so in theory, these waves don't communicate with each other as well, right? So those brain regions, if they are coming from two different brain regions or two different populations of cells, they'll communicate less strongly than A and B will. And these indicate structured patterns of excitation and inhibition. That's the key idea here, is that we know that neurons fire action potentials, right? But what drives these phenomena are neural oscillations to a large degree. You need to have this temporally refined control of excitation and inhibition. These on and off states have to be controlled very systematically, and oscillations or brain waves are the way that this is done. And so we'll call this phenomenon coherence to make things simpler, right? The phase here is the key. Our second goal here, now that we understand a little bit about what brainwaves are and how they're generated, is to understand how brainwave synchronization might play a role in cognition and behavior. So the first instance that I feel is relevant, the most classical example of coherence in many ways is to depict corticospinal activity, like just a simple movement of your hand. And so and we all know about the cortical spinal tract, the connections between the brain and the spinal cord and your muscles, right? So we know that here, for example, so they use MEG in this study, and they read the magnetic signals from the surface of the scalp, and they attached myoelectrographic, right, electrode on the muscle in the hand. And at the time of a movement, at coherence between the electrical activity observed at that contact on the hand and the magnetic, which is also half electrical activity observed on the scalp, those two different patterns of activity are synchronized in this frequency band, this 40 to 60 hertz there, during movement. And so this is a classic example that just shows how coherence of brainwave synchronization is relevant to behavior, because the simplest behavior of moving your hand involves an increase in coherence between motor cortex and the neurons that cause your muscles to move. So this suggests that there's some sort of strong link in terms of synchronization between brain areas. And so the information is likely conveyed via that mechanism. Coherence also predicts reaction time during focused attention. This is a study we did using EEG, electroencephalography. So we just put electrodes on the surface of the scalp and read the currents. And we use source localization to find that these two brain regions, their coherence, their synchrony, predicts reaction time in a cognitive control task. Cognitive control is just focused attention. So if I ask you to attend to a specific object that has many changes, and you have to attend to these different changes and respond effectively and quickly. Whenever we give people tasks like that, it's challenging. And so these parts of the brain that are involved in attention are activated. And so their co-activation or their shared phase or coherence predicts reaction time in this study that we published recently. And the third component here is that coherence can integrate sensory information. So by sensory information, I mean sights, sounds, colors, things that evoke our sense perception, touch. And so for this, they commonly, very similarly to the previous study, they inserted an electrode in the auditory cortex of a monkey, and they stimulated the median nerve, which is a nerve in your hand, right? In the monkey's hand as well. And so stimulation of that nerve caused a greater response in auditory cortex. So if you simultaneously stimulate that nerve and play a sound, you get a bigger response in this bimodal condition, meaning two modalities of perception. You increase the response in that area with bimodal stimulation. Whereas with somatosensory stimulation alone, which is that final box, the third one to the right there, you see there's no response in the area to just the stimulation of the medial nerve. So synchronization between multisensory areas and primary sensory areas like the auditory cortex are presumably the mechanisms that support this kind of increased response to dual sensation events. Because if you imagine the world that we live in, a lot of times the sights and sounds are happening at the same time. So you get multisensory integration just effortlessly. Traffic lights are often, when the light changes, they are often accompanied by changes in sounds in the environment, the sounds of engines revving up or the sounds of brakes or things like that. So our brains are very specially designed to integrate this kind of information very quickly. And presumably coherence is a key mechanism for this kind of phenomenon. The fourth example that I want to show you is that coherence can link the brain regions that underlie our emotional experience in life. And so we've all probably heard of the limbic system, which has a part of it as the amygdala and the hippocampus. And these two regions are important for emotions and fear and memory, respectively. Remembering what happened to us in the past, whether it was good or bad. Now, synchronization between these areas can predict mood scores. Memory as well in other studies. But here we're talking about mood. And so lower coherence between the hippocampus and the amygdala predicted mood in this study. And so which suggests that an increased synchronization between the highly well-known emotional center of the amygdala and the highly well-known memory center of the hippocampus. Greater synchronization between those areas predicts worse mood. Now, what does that suggest? That suggests, to me at least, that increased synchronization suggests more information content being shared between this memory and an emotional region, which probably does predict, and we can see here in this line graph that it does predict their mood across many patients.